On behalf of the Indian Academy of Sciences, I have great pleasure in welcoming you all to the 28th mid-year meeting of the Academy.
I am close to the end of today's session. I do realize that your gray cells have been taxed sufficiently, but let me tell you a little bit about the story that I have been indulging in for the last 25 years. So this really is a disease that I've been working on, and as a medical doctor, and I think all of us who are in science over here, we all have one common theme when we start working or doing research, is that we want to make a difference. It may be small, but we have that little dream in us, and I think that keeps us going. So this is really why I chose this particular disease to work on, because I could tell you what is happening inside the human body. So it basically falls in the category of neglected tropical diseases, and as you can see, the newspapers cover it, that India is facing a heavy burden of neglected tropical diseases. So note that it's not just a tropical disease, but it's even one shade lower, it's actually a neglected disease. And amongst these neglected diseases is one particular one which heads the list, which is called Post-Kala-Azar, which has been with us since the early 1800s that it's been with us. So this is the disease that I've been working on. We're coming close to elimination, which is really exciting, and I'll tell you how we're playing a part and how we're able to hopefully make a difference. So if we move forward, then we're now at a point, we now come to 2016, where we say that we're almost there and we're close to tipping point. And why do we say that? We say that because the government also has taken this up, and in the health budget of this year, there was an emphasis that 2017 will be the year that we will be able to eliminate Kala-Azar and filariasis. Unfortunately, politicians like to say a lot of nice things, but let's see how close we are to the truth, whether 2017 we'll be able to do it, or we'll ask for a few more years to get there. And what are the limitations of not being able to get to 2000? We are already in the middle of 2017. So basically, what do we understand by Kala-Azar? As the name suggests, Kala means black, and Azar actually in Bengali means fever. It means jor means fever. So this was a black water fever which was occurring in the eastern parts of India. It's called visceral leishmaniasis because it affects the body. Just to take you, because there may be some of you who are not familiar with the life cycle of the parasite, but all I need to tell you is just two things we need to know. Is one is the human is the host, and the vector is the sandfly which is here. So the sandfly infects the human, it enters into macrophages where it proliferates, multiplies, causes enlargement of the liver and the spleen, and then when subsequently when an uninfected sandfly again can take a bite, it will again multiply within the sandfly, and then it will come up to the midgut, again bite an uninfected individual, and so this vicious cycle continues. All we need to know is that man is the reservoir, and we need to know that these parasites live within our macrophages, which is within the reticuloendothelial system, which is mainly the liver and spleen in our body, but basically macrophages, which may be in the skin, but may also is usually in the liver and spleen. Okay, so a little bit to go backwards to know where we are today. So the enemy, when we started knowing about this disease, we actually did not know. The enemy was unknown at that point, and it was in 1880 that a mysterious fever started in the northeast, basically in the Garo Hills. And because it was a fever, and at that time malaria was already known, They called it, they didn't know what to call it, so they said this was a quinine-resistant malaria which had appeared in eastern India and had appeared in Dum Dum, which is actually North Calcutta, which is where the airport is. So if you know this, you'll be familiar with this name. So they thought this was just a variant of malaria which was occurring, but subsequently this fever spread to a large number of areas, and there were huge epidemics which occurred, and many people lost lives over there. Fortunately, we did find a drug which appeared at that time. So this was at this point that the enemy was unknown. Fortunately, due to the help of two excellent pathologists, one who was William Leishman and the other who was Charles Donovan, both of them reported independently that they had identified in patients these bodies within the autopsies of the liver and spleen. These are the macrophages, and these little dots that we have are what we call Leishman-Donovan bodies, okay? So Ronald Ross was responsible for calling them the Leishman-Donovan bodies to honor both the pathologists who actually identified them, and they did it independently, both published in the British Journal of Pathology in the early 20th century. So coming forward, so as we can see, it actually has a pretty substantial global burden. There are 98 countries which are affected. It may affect the skin, in which case it is called cutaneous leishmaniasis, but more important for us in India is the fact that we have the enlargement of the liver and spleen, which is why it's called visceral leishmaniasis. And this visceral leishmaniasis, in a very small proportion of individuals, 10% of these individuals will develop a skin disease which is called Post-Kala-Azar dermal leishmaniasis, okay? So that's why it's called PKDL, and I'll stick to PKDL rather than continuing with the large name, okay? So PKDL is what we need to focus on. And why is PKDL important for us? The reason it's important for us is if I showed you, this is the entire burden that we have of leishmaniasis worldwide. But look at the burden of visceral leishmaniasis. It's much, much smaller. And look at it, the only places where you get PKDL is two areas in the world, okay? So India and East Africa and Sudan are the only two places where you will get cases of PKDL. So you can tell me why bother about it. There are just a few cases. I'll tell you why as I go along. So as you can see, West Bengal, where I come from, is a major pocket where it occurs, but Bihar, of course, is the largest pocket. So this would be a patient of Kala-Azar that we have, and these are the number of individuals that have come to us with various symptoms of PKDL. Basically, they would have pigmentation, loss of pigmentation of their skin, or they would have small nodules which appear over them. I'll show you a few more which are actually nastier, but just to make the point, drive the point home. So the reason why they've been thinking of elimination for a long time, and why the government has said 2017 is the target, is the fact that it has such a small geographical distribution. It's only in 4 districts in 4 states in India, and only 54 districts in India. So it is achievable, definitely it's achievable. So that's why they've considered elimination and subsequently, of course, eradication, like we've done with polio and like we did many years ago with smallpox. So it's slowly coming into that category. But are we there or not? We unfortunately have a series of delayed targets for various reasons, and I think I have been able to pinpoint the reason. So in the early mid-90s, early 2000, there was a substantial decline. So people at that point said, okay, we can think of elimination. This was 2003. So in 2005, what happened was, there was an agreement between the main countries where Kala-Azar occurs, and they said, let's try to bring it down to 1 case per 10,000 per Jilla by the year 2015. So that's 10 years later. In between, in 2010, another problem occurred, is the drug which was being used, which was sodium antimony gluconate. There was a huge degree of resistance which had appeared. Almost every second patient of Kala-Azar did not respond to antimony. So this added to the burden, and we have a very limited repertoire of drugs which are available for this disease. Come 2015, where did we stand? We still were nowhere close to elimination. We had brought down the numbers definitely, but at this point, that's when they woke up and they said, yes, possibly we need to attack these cases of PKDL, because these are individuals who are roaming around within the community, do not have any disease which demands treatment. So they basically are individuals with skin lesions. The sandfly takes a bite from them, goes and affects uninfected individuals, but they will not themselves seek treatment. And I'll show you some cases and I'll tell you about it as I go along. So this was a game changer where they said, okay, let's identify cases of PKDL as a potential disease reservoir. We've come to 2017. We are doing very well, but the next target that we have is 2020, where I think we will be able to say that we are close to elimination, and then we need to start thinking about eradication. So the important thing was the identification of PKDL, okay? So if I look at the number of cases, so what we did is the government encouraged this basically. It was taken up in West Bengal predominantly, that we went on a door-to-door survey to patients who had visceral leishmaniasis, and examined them and said, do you have any skin lesions? And I'll show you the numbers as they appear. So what happened is, this was in Bihar. You can see the number of cases of Kala-Azar have come down substantially, but look at the number of cases of PKDL that have gone up. This is a smaller scale, so don't compare this with this. This is a smaller scale, but it's just to highlight the fact that the numbers have increased, okay? So it's not that more cases were occurring, it's just that more cases were being detected. What about Jharkhand? It was exactly the same scenario. It was exactly what was happening, the same thing was happening. What about West Bengal? West Bengal actually, if you see, we started in 2014. So in 2014, you can see, 2014, 15 and 16, we've been doing this active door-to-door survey, and we've actually been able to identify cases, get them to seek treatment. We have them coming. I'll show you what we do. So we make them come to camps, we help them to diagnose them, and we have to motivate them to take the treatment, which is not the most pleasant of treatments. So this is the important thing, the fact that we hadn't identified all these silent cases which were there, all these basically which were hanging around in the system but were not seeking treatment. These were the individuals who are going to be the major obstacle as far as elimination is concerned. So what are we doing about it? So as I said, these are the patients, these lesions are absolutely full of parasites, okay? So just to tell you, how do we identify, what are the ways by which you can identify is the disease reservoir? This was one of the first cases that he came, I think he came in 2005 to us, and he had all these lesions over here. And so I asked him for how long. I think for 8 years he had had PKDL. And I said, why have you come after 8 years to get treatment? He said, I've only come because I'm going to get married. That was the only motivation. He didn't think this was cosmetically acceptable. That's the only reason why he came to us. Fortunately, what we did was we just took a sample from here, put it into some tissue culture medium, and in 24 hours, they were absolutely full of promastigotes. They were just teeming with promastigotes. So these are basically full of parasites. So if you don't take care of these cases, you're never going to be able to eliminate PKDL. The other way that you can do it is actually prove it by a system called xenodiagnosis, where you actually have a case of PKDL, make the sandflies sit inside this little thing, and then collect the sandflies. And this has not been done by us, but it's been shown that essentially it was actually convincing the scientific community that yes, PKDL is the disease reservoir. You can eliminate all the cases of visceral leishmaniasis, but if you don't eliminate these, you're going to be nowhere. So then we went forward. We said we do believe that elimination is achievable because we know that man is the only host, sandfly is the only vector. We do have good rapid diagnostic tests, not for PKDL yet. We have effective drugs available, again for VL, not for PKDL, and importantly, the geographical spread is limited. And very importantly, there's a very high degree of political commitment. So basically, as I said, it's these two areas that I'm going to be talking about is where we have the major stumbling blocks. We don't have good diagnostic tests yet, and we don't have effective drugs. We're still at a point where we don't know which drug will work. They haven't been carried out. The studies have not been carried out. So this is where I work. This is the city of Kolkata, where this is Dum, where Leishman and Donovan did their work. This is the School of Tropical Medicine, where we get our patients. And I'm located in the middle at the Postgraduate Medical Institute, where actually Ronald Ross discovered the transmission of malaria by the Anopheles mosquito. So this is taking you to a city in Peru, which is in South America. And this is possibly the world's first botanical laboratory. And what's interesting here is that these are different geographical zones. As you go along here, they identify different geographical zones, and this is the way by which they studied which seeds would grow in which climatic zones. So basically, this was a large laboratory going back to the Incan civilization, which was probably in 1500 or 1200 AD that it was there. So my research group is on very similar lines of thought. We have an immunopharmacology group, and the areas of interest that we have is, the first one we've been doing very actively is diagnosis and monitoring of chemotherapy. This is Srija and Shushmita who work on this. Another area that we have been working on is immunopathology of the disease, because as I said, there is no animal model for PKDL. So the only source is the PKDL cases itself. So in vivo is the only way you can verify that what is happening within this disease. So this is Sushmita, this is Shivabroto, this is Debanjan, and this is Shudipto who work on the immunopathology. And the last but not the least is the group who works on chemotherapy. I won't be talking about this work. So this is Gautam, this is Rupashri, this is Obijit, this is Piyu, this is Obijit again, who are doing this work. So coming into where we are basically working, what are the challenges that we have? The main challenge that we have is we don't have an animal model. Because we don't have an animal model, you are exclusively dependent on whatever you get in these patients. And remember, there are only 2 zones in the world which have this particular disease. Disease of the poorest of the poor. They really come from the poor, really are extremely poor. The government has a system by which they give them a little bit of money. I'm nowhere near finishing, but anyway, so let me get quickly. So let me tell you basically what we've been doing. So what we've been doing since December 2014 is carrying out a door-to-door survey essentially. And what we do is we get the cases that we have, they come, so we get the samples over here, and then we pick them up, and then we do a PCR, and then we do a qPCR also to be able to do the diagnosis for this, and then based on that, they receive the treatment. So how have things been looking? So the laboratory diagnosis that you have is basically the clinical diagnosis, you have the antibody tests, and then you have the antigen tests. So this is the one that we've been focusing on. Where are we at the moment? If you look at the PCR-based diagnostics, this is a very simple method where we have a positive control and we do it using the ITS1 PCR. This is cases of PKDL also that we've picked up, and we also have a method by which we actually quantify the parasite load, because that's important for us when it comes to treatment. We want to know whether the treatment is working or not. So this is the load that we have in the different ones. So it's quite a substantial parasite load that we have. What's important is the treatment that we have. The treatment you have is 2 possibilities. One, you can get miltefosine, and look at miltefosine. Miltefosine brings down the numbers from 53,000 to 38. So we're doing very well as far as miltefosine is concerned. The other drug that you have, which is very effective, it needs only a 3-week treatment, this is a 12-week treatment, is amphotericin B. Amphotericin B, unfortunately, has a substantial number of parasites which is existing, and that's really bad news, because A, you're not treating them, you're sending them back with parasites still there, and B, there's a chance of resistance developing. So 6 months later, the miltefosine group did very well, the amphotericin group did very badly. They came back to us and there was a resurgence of parasites. So this is where research comes into play, because nobody so far had actually measured the parasite load. People thought that 3 weeks of liposomal amphotericin B was perfectly fine. So once we showed this, the government woke up, and the government has now said no more of liposomal amphotericin B, just stick to miltefosine till we find a better modality. The other aspect that I'm going to be talking about is the immunological aspects, is where the parasite essentially lives within the macrophage. And to live within the macrophage, it comes up with a nice understanding. I call it a marriage of convenience, because the parasite normally, if when you have an infection, what will the macrophages do? The macrophages, or the antigen presenting cells, will essentially get activated. They will break down the parasite or microbe or whatever it is, and present it to T cells. The T cells then will get activated, and finally there will be elimination. But here what the parasite is doing is coming up with an agreement and making them alternatively activated. And by making them alternatively activated, it doesn't allow antigen presentation to take place, and you end up having a proliferation more of the T helper type 2 type of cells, which allows these cytokines to increase, and essentially what you have is an immunosuppressive milieu, and there is parasite persistence. So what we wanted to see is, okay, all this is very well, but what is the story as far as PKDL is concerned? This is all being derived from animal models of visceral leishmaniasis, or patients, very few with patients, but mainly with animal models. As I said, there is no animal model for PKDL, so we needed to see what really happens. We did find a decrease in the most important antigen presenting cells in the skin. So this is basically the CD1 cells, and you can see you don't have them on presentation, and they reappear as when treatment has been completed. So there's a depletion of the most important cell which is necessary for immune functions is the antigen presenting cells are impaired. There's a total absence of CD4 T cells. There are no CD4 T cells which are going to be there to be activated and things. There's a huge infiltration that we have of CD8 T cells, but most importantly what we had is an increased presence of CD68 type of macrophages which were present. Now these macrophages had to be alternatively activated for them to be able to ensure that there is an immunosuppressive milieu. So how do we check that? Normally when you look at macrophages, when they are exposed to a microbe, they will be a classically activated macrophage, and you will have all the typical markers up. There will be increased amounts of microbicidal molecules, which is reactive oxygen species and nitric oxide, so as to eliminate the microbe. But the parasites are smart guys. They ensure that the parasites get alternatively activated. They ensure that the parasite killing molecules have been removed. They are decreased. These should be upregulated. These are markers of alternatively activated. These are regulatory cytokines that we have, and you have the vitamin D family. So we looked at all these markers. I'm not sure what the time is like, but anyway, yeah, I'm done. All right. So let me just put it together. Basically, what we did have is an enhanced expression of arginase. We did have an enhanced expression of mannose, and we did have an enhanced expression of vitamin D. But so basically what I'm trying to say is that we did show that they were alternatively activated. So the take home message I have is active surveillance is essential to be able to treat PKDL, which is critical for elimination of leishmaniasis. The parasite very cleverly generates an immunosuppressive milieu, so we need to generate immunomodulatory drugs which will have both parasiticidal activity, kill the parasite, as also modulate the immune system, so that to be able to ensure parasite clearance. So thank you very much, and thank you for your patience. These are my collaborators who've helped me bring this story to where it is.
Patients who have had
So patients who've had Kala-Azar, 10% of those individuals develop post-Kala-Azar dermal leishmaniasis. People don't know. We don't know why 10% of individuals will actually develop it. So that's still an unknown question as to why some individuals, 90%, are cured of VL. They don't develop PKDL, but 10% move on to PKDL.
Yeah. Does PKDL always have those nodules?
Yes, it will have. There are three types of lesions. You'll either have just depigmentation, like you have in individuals who have vitiligo, just depigmented patches which are there, which are very diffuse, or they'll have small nodules all over the body, or slightly raised, or what we call papules. So basically, it's a little bit of elevation of the skin which takes place, but it's very diffuse and it's very diverse. So
How do you link arginase overexpression with this?
Okay, so arginase basically is, as we know, that arginine can either by inducible nitric oxide synthase get nitric oxide will be produced. Okay, nitric oxide and ornithine will get produced. We don't want that. We don't want nitric oxide because nitric oxide will kill the parasite. So what it does is it pushes it towards arginase so that you don't have nitric oxide and instead you just think. So that's why. So that's important for parasite survival.
In your experience as a physician,
yes,
did you find this disease communicable that easily?
It is communicable in that the sandfly can take a bite from a patient with PKDL and can bite others in the community. So when we did a cluster study, when we did this house-to-house survey and we tried to identify what were the cases, we have found that the cases of PKDL, they tend to live in clusters. It's in one household, not all will be affected. So all don't get affected.
What is the source of your optimism that this disease can be eliminated?
By just going door to door and picking them up. You just have to pick them up, follow them, monitor them, and pick them up. The numbers are very few. The numbers are very few, so it is possible. It is possible. The numbers haven't increased. So over a period of time, we've had 200. So 200 patients, you can actually monitor them. Or the cases of Kala-Azar that we have, if there is active monitoring, because you see, as I said, it's a political commitment. If the government gives you Kala-Azar supervisors, you will be able to identify these cases.
Do you have the same hope for Tuberculosis?
Tuberculosis is a bigger challenge. Now see, we said 2025, but that's because drugs are a problem. It's a longer treatment. The chances of drug resistance are much higher. So the challenges are much greater in tuberculosis.
Madam, is it also possible that some person say he never developed Kala-Azar but contacted this Leishmaniasis and PKDL?
Yes, very few. You see, we're working with a community where the educational level is low. So often you may have had it as a little child and you don't know. So the question that we ask them is, we don't ask them whether they had Kala-Azar, but we ask them, dawai suya mila tha ki na? Because for 20 days they get injections of stibogluconate, and that you don't forget. If you get 20 injections in your life every day on your, you will remember it. You will remember that for sure. So that they remember. So we never ask them bukhar hua tha ki na? We ask them suya mila tha. Then they say haan mila tha. Bahut. So you have to tune your questions based on the people you're working with.
Ma'am, I have a query regarding its investigation using PCR. That you are taking the tissue culture sample or the?
We are taking a skin biopsy. We take a skin biopsy. So wherever the lesion is, we take a skin 4 millimeter biopsy from there.
And the total DNA has been extracted.
And then we make the DNA. So mainly it's genomic DNA. For the parasite DNA, we amplify.
Okay.
We amplify the parasite DNA.
The specific gene you are amplifying?
Yes, it's a kinetoplast DNA we're looking at. I'm sorry, I ran out of time actually. Thank you very much. Thanks a lot.